So I'm an epidemiologist here in Bristol and the theme lead. I'm currently on sabbatical in the German Cancer Centre in Heidelberg, and just so I don't have to tell everybody over coffee where it is, describe one of my German friends as the Cambridge of Germany, in the southwest of Germany, so if I can work out the pointer, Berlin on the east, Heidelberg there just north of the Black Forest, close to the French border. It's on a lovely wooded valley of the River Neckar, which feeds into the Rhine that arises in the Black Forest. It's a small place, but has a lot of visitors. It's actually the oldest German-speaking university within what's currently Germany. There are older universities, but they're no longer German universities. And it's not bad in the world rankings, and it's a lovely place to be. So enough of my gloating. What I'm going to try and talk about, in a way, it's surprising that Bristol is really good at nutrition, because I don't think it's that visible. We're top 5 in the metrics. And so I'm going to try and tell the story of why that is and all the great stuff that's gone on and how the BRU and the BRC can bring that together and strengthen it further. So I'm going to start from nutrition and health and in a sense of population perspective. I'm then going to define clinical nutrition and try and pick up the threads of clinical nutrition research over what I call past, present and future, and I'll define my terms as I go. So, nutrition and health. I always like to start with comparisons. So this is some old data about hunter-gatherer diets. We, for many years, or much of evolutionary time, were hunter-gatherers rather than farmers. And there's pretty good evidence that actually the first farming communities, the diets represented a nutritional nadir in human evolution, that they and that the farmers were shorter, they potentially less healthy. And don't get me wrong, being a hunter-gatherer wasn't brilliant. If you got hurt, you didn't live a long time. But potentially it's the diet that in some ways we're physiologically adapted to. And striking things about it, this is a comparison between hunter-gatherer diets and US diets. They didn't eat a lot of fat. They ate meat, but it would be wild meat, it would be low in fat and low in saturates. They also ate a lot of nuts, tubers, berries, and so vitamin C as a marker of fruit and vegetable intake was high. What they wouldn't be eating was dairy produce. They didn't know how to get the lactose out. The adaptation, as I'll show in a second, came later. They wouldn't be eating much cereal fibre. So this idea that this healthy diet or healthy breakfast, and we'll talk about breakfast in a second, this healthy breakfast would have been your cereal fibre with your natural yogurt on top. If you're thinking in evolution terms, perhaps not. Why it's relevant today is people are very interested also that hunter-gatherers potentially were hungry some days. So these 5-2 diets, Paleolithic nutrition, the effect metabolically of fasting, and it's something that we're touching on and other groups are as well. So there's renewed interest in this. I just wanted to show that the adaptation to farming was relatively recent. This is a graphic from Nature, and as I was pulling it up and reading it and just checking that I knew what I was talking about, which at times obviously you may worry about, but basically around 10, 11,000 years ago, first of all we learned how to deal with milk, and then the adaptation as well comes to lactose tolerance, so we persistently can metabolize lactose. And I actually found that this work, these are fragments of pots that we use to process cheese to separate curds and whey from burial sites. This work was done in Bristol. So this is done in our Department of Chemistry. And maybe one of the challenges going forward is to link in everybody who works in nutrition in some way in Bristol into the BRC. And we have tried before, but it's something that we should move forward on. So other comparisons. So I thought, as everyone would likely have had their breakfasts, I looked on the web for traditional breakfasts. So this is the USA, Greece, and Japan. Apparently this was quite controversial as a picture of an American breakfast. Some people said, where's the rest of the bacon? And then there was contention about whether it was the right sort of breakfast handgun, because... apart from the gun, you'd be pretty confident this would be your full English this is supposed to be a picture of a Greek meal. And in many senses, what nutrition policy in the UK is to Mediterraneanise our diet. And this then is our picture of a Japanese diet, so with rice at the centre of the plate. These are some descriptions of these things. So this is the traditional Japanese diet. So this is our big contribution of rice, a lot of fish, permissive about alcohol. Meat is really a garnish and not a high fruit and vegetables. This is the meat and 2 veg. So I don't know where the veg was on this breakfast. It's probably just out of view. But it's very much meat at the centre of a plate of fish maybe on a Friday. And then the Mediterranean diet, which is fruit, vegetables. And it's not the Mediterranean diet, it's the Mediterranean diets that have some common features that are common to around the Mediterranean Sea. And to some extent, this is also their summer diet. And there's issues about whether optimising health and when you look at Greek migrants who go to Australia, perhaps what they do is they eat their summer diet all year round, and that's why, despite the fact that they put on weight, they eat more meat, they smoke, they actually do better. And that's really, if you think about our dietary advice today, what we would tell people to do to become more Mediterranean. This is the life expectancy in people aged 45, so they're not huge differences. And the reason for having the handgun in there, apart from a sort of cheap jibe at Americans, was also to illustrate that these ecological comparisons, there are other ways to die apart from the food that you eat. So when you do these ecological comparisons, there are other differences between societies. The big thing, and I think this is one of the mistakes that a number of epidemiologists made in the last 20 years about diet, and George was vociferous in pointing out how socially patterned diet was. I think people made great strides when they set up big cohort studies. They went away from case control studies, said, no, we want to get rid of bias. We need to do studies that are large enough. We need heterogeneity of dietary exposure, so they set up the EPIC cohorts. But what they discounted was the fact that people who are more advantaged and less advantaged eat different diets. So this is some UK data. You're getting big differences in how much fruit people are eating, how much vitamin C, and it's very hard to tease out those differences. I used to be taunted by George about my PhD, which was on vitamin C. He said it's just a very good marker of what social class you're in. But to some extent that's true. And the proof of it here, what I should say is that I'm shamelessly self-citing here, so if these are just 2 examples from things that we published, but many people did similar graphs. These are the ideas of magic bullets making a difference. This is vitamin E, this is a meta-view. These are a series of observational studies where we asked people how much vitamin E do you eat and related it to cardiovascular mortality. Observational studies seem to show vitamin E good. You do a series of trials of vitamin E where you randomise people. The only difference is what they got allocated to receive. Vitamin E, thankfully, makes no difference. At least it doesn't kill you. And in the case of beta-carotene, it also was turning people yellow, so it was doubly great. And again, with vitamin C, there was lots of hype about this. With antioxidants, we just found some of the smaller trials, even before the large conclusive trials came out, there was really precious little evidence that vitamin C was going to make a difference. So based on observational data, we were pushing forward with these magic bullets, and they've been disappointing. So we need to rethink how we draw causal inferences, how we find out about diet. And in the end, the danger with all of this is you come away saying, well, I can't say what a healthy diet is. I don't think that's true. I think I could, in general terms, point to you and show you and consistently. I never think this is a particularly sort of exciting prospect as your lunch, but this is very much Mediterranean. We're pushing fruit and vegetables into a bigger chunk, meat and dairy getting smaller. This is our carbohydrates. And maybe that's looking a little bit more appetizing as a Mediterranean diet. But that's, in general terms, I'm quite confident about that. What I struggle with is how I would do specifics. I've always championed the idea that we do food-based trials. The problem with this is that we don't necessarily blind people. So we don't know if by getting people to eat one thing, they also do other things, change other things about their lives. We also need to convince people in the general population to change their diet for a long time to achieve sensible differences. So hopefully, and that's the link with population science, epidemiologists can get a bit smarter. So being aware of confounding, using novel approaches, and I'll show you a couple of examples, and also using omics type approaches to draw causal inferences. So we can get closer to the specifics and then test them in trials and in clinical populations. So now I begin the talk. So I want to talk about clinical nutrition. So that's the general preamble about nutrition and population nutrition. And first of all, I'll define my terms. So Wikipedia obviously is a source of all wisdom, but also the European Society for Clinical Nutrition and Metabolism. Clinical nutrition is nutrition of patients in healthcare. Clinical in this sense refers to the management of patients. And that's very much what we focused on in our nutrition theme and in the BRU that preceded it. So that's what I'm going to try and draw out from the nutrition work that's happening in Bristol, is how people have been working in clinical nutrition and how it's changing. And why is clinical nutrition so exciting to be involved in? One is that just as everywhere you look, there's limited evidence to inform practice. You say, what do you do? And someone says, well, we do this. And you say, why? Well, a few of us got together and had a chat and discussed, and most of us think this is a good idea. But the evidence to inform practice is not great. We obviously have examples where it's important. This is phenylketonuria. So where we put, everyone has their heel prick test, and we put people on a low phenylalanine diet, and we make a big difference. But we've got little other areas where we would actually be clear that dietary interventions in clinical populations make a difference. But potentially this is a more motivated, identifiable group. I've put a question mark there because I'm not sure it's always true. I think we struggle with type 2 diabetics to motivate them. I think to get them to lose weight, to become superactive is a difficult thing. But generally, clinical populations are more willing to engage in things. We can therefore potentially achieve big differences in the diets because people are motivated. We're looking for effect sizes that are clinically important. So we're not looking for something minuscule where I need many, many thousands of people potentially. I'm looking for things that make a difference to people's lives. And I can often measure that difference using some short-term outcome measure over months rather than waiting years to see what happens in terms of mortality. So clinical nutrition research is fantastic fun because you can potentially do a heck of a lot of good. So how did this come about in Bristol? And I think there's a series of threads. What I'll start with. So this is a very Ness-centric view of nutrition. So past, present, and future represent the time I've been in Bristol. I came in 1997. The past is the time before we had the BRU funding. I've called the present the BRU, and the future is the BRC now and beyond. And a disclaimer, this is me picking things that I'm familiar with, shamelessly self-citing. And I was talking to Ashley about it just before I started the talk, and he said he was going to count up how many times I mentioned him, so that's one. But if I've omitted people, if I haven't quite done your studies justice, trying to give a broad brush of 20 years of work in an institution like this is a challenge. And I think the things that are fed in, I've got children and pediatric research. I've got exercise, nutrition, and health sciences. A thread around perioperative and surgical nutrition, epidemiology, health services research, and then the nutrition behaviour unit. And I'm going to work my way through those and just give some examples of the work that was being done, and then some that's fed into the work we've done through the BRU. I've put ALSPAC under children and paediatrics. It's clearly population science, but it was initiated from the clinical paediatric department, and I'm sure people will contest that. It was certainly used a lot by people in the Centre for Child and Adolescent Health, looking at things like breastfeeding algorithms for growth, anemia, when you should treat. And then I've got hospital-based paediatrics, which I'll come to in a second. So this is, and in Bristol, when you start talking about anything, it's very hard not to now talk about ALSPAC. So it's our started as prospective birth cohort, or a prospective cohort of pregnant women. It was, I think, initially the Avon longitudinal study of pregnancy and childbirth, and it's evolved to this, which actually is a title that's stuck. There's a South African birth cohort that called themselves Birth to 10, and then as they got some more funding, became Birth to 20. So I think this was a better choice in terms of renaming. And the kids now who are born to those pregnant mums are some 30 years on. And the reason for bringing Auspex up is, one, it's the jewel in our crown of our population science studies, but we started putting important nutritional data into it. So one of the things that we achieved was to put in DEXA data. So this is one of the first studies of, this is the FTO gene, so it's people are essentially finding common polymorphisms of small effect. Lots of studies showing associations, modest associations. So even though this is the, I just had to see Tim Freding holding up the difference in the amount of lard that you get with the gene. The differences are fairly modest, but the nice thing in Auspex is that we had DEXA data, so they could actually look at the effect with fat mass and the effect with lean mass. And actually having this richer nutritional phenotype actually makes, to me, it more compelling that it works for fat, but not really for lean. So then picking up hospital pediatrics. So this is a trial that Julian Hamilton-Shield led around speed of eating. Julian set up an obesity clinic, got 106 obese young people, used this machine called the Mandometer, which essentially is a scale that you put a plate on, tells you how quickly you eat, and you can retrain how quickly someone eats. So the idea is that if you eat slowly, you get a better satiety signal, and you actually end up eating less. And this is just a scatter plot of the data in terms of the SDS. On an average, it was a reasonable difference that was maintained at 12 months. And I'm not going to explain these in detail. Julian knows because I don't really understand it. But what they also showed was that they reprofiled or retrained the hormones. So you're resetting people's hormones when you're resetting their eating. It's been a challenge, actually, to roll this out. In an obesity clinic, you've got people who recognize they've got a problem, coming forward, and they are seeking a biomedical solution. This works. When you try and take it out into groups of people in the general population, it has been a challenge, and a large trial that Julian and others tried to recruit to struggled. And this is a real challenge for us of how to bring people with, young people and older people with obesity in and motivate them and get them to change their diet. But I think this has been an important thread in terms of shaping nutrition research. Some recent studies I'm not really going to allude to because I think they're going to come up with our PhD students. We are doing MRI scans. We are using glucose monitors. Laura's going to give her a tape talk from South Africa that she recorded. She shows this trace. This is a child with cystic fibrosis diabetes. She undersells herself. This is her best trace, obviously, but this is the same child with her low glycemic index diet. She's really shifted these curves down. Now, maybe that was a special child and we can't actually get it to work for everybody, but it does look promising, and people just haven't taken groups like this and worked out what works, or if nothing works, at least we can say confidently, we tried. So this is exercise, nutrition and health sciences. And I was 20 years ago when I just arrived on Senate, and it was just being renamed with the nutrition added in. So it was a big thing to rename a department back then. This is what they say about themselves. We conduct research that focuses on physical activity, nutrition, and their associations with health across their lifespan. International reputation for teaching in physical activity, nutrition, and community health. I put that in just so that I was sure that I'd got it correct in terms of what they say. My summary of what they do is to work in free living subjects. We're not like Leicester Loughborough, who have huge gymnasia testing facilities. They do sports science. Fantastic. It's not what we do or where our expertise lies. But when it comes to studying people, free living subjects, Bristol is up there. We've developed better and novel assessment measures. And a large clinical trial early acted, I think, has been big in terms of shaping the work going forward, which has led us on to some of the sedentary work. So I'm just going to take us through that. This is some work actually done in Alsbach with Chris Whittaker and I did. These are movement sensors. These are uniaxial movement sensors you put around the waist. We were some of the first groups to do it on thousands of people. Most of them came back. This is one that came back via the dog. But most were returned. We could give them a trace. And this is what transformed physical activity measurement. We can actually see what someone was doing minute by minute. And this is the association with obesity. We can see stronger associations cross-sectionally. It's a weaker association when we look at it longitudinally, and that may be because we were more naive about the role of reverse causality, that some of the association between obesity and physical activity goes the other way. So if you get fatter, you become less active. But I still think that in part physical activity leads to obesity. This is the early active trial, which was led by Rob Andrews and with Ashley as part of the team and others in Bristol. A trial of people with new diagnosed type 2 diabetes. 3 arms, usual care, diet, or diet and exercise over a year, with the primary outcome of HbA1c, so it's conducted in the southwest. And in terms of changing behaviour, so we've got our 3 arms. The usual care, not much change. This is moderate and vigorous physical activity. So the sort of activity that gets you out of breath, makes you a bit sweaty. This is diet and exercise, so the exercise didn't increase it, but we're looking at only a few minutes a day. Diet didn't make much difference. In terms of weight, diet and exercise and diet had similar effects. And when you look at changes in HbA1c, so a measure of glucose control, you see that actually diet and exercise and diet do similar things. So it could be that we're nihilistic that exercise just doesn't work, or actually that we weren't really able to change it, or we have to get clever about how we change activity. And some of the work that Ashley's led on over the last 5 years in the BRU has been about exploring sedentary behaviour. And I was always very sceptical about this. I just thought you had to be tortured in lycra to get any sort of benefit, that just to get up and walk around a bit, come on, it can't be that easy. But actually we've got some nice studies showing that actually if you sit people down and get them to walk around a little bit, we can make changes to their glucose curves. And actually we can begin to map out how people spend their days, how quickly they're moving, when they're doing moderate and vigorous physical activity, when they're not, and identify the opportunities, resulting in sort of attempts to improve the health of overweight men and women using electric bikes, and we're going to hear about the project that's going on with that. So it's been really interesting that we've ended up giving sedentary bikes to people with type 2 diabetes. Another thread is perioperative and surgical health. So in the bad old days, potentially someone, when they were coming up for an operation, they'd be fasted for a long time before and afterwards. And people started saying, well, hang on, does that actually make a difference? And people had started to do randomized trials, and here in Bristol, they pulled together that data. This is Stephen Lewis, who's based in Plymouth, who still works with us, and others in Bristol. And the suggestion was that on a number of outcomes, apart from making you feel sick or be sick, it actually, if anything, was better. So instead of actually not feeding people, probably we should be trying to feed people when they're ready. One of the issues is, yeah, people do get sick, and they're not super happy about that, and neither are the nursing staff. So one idea is that we could sham feed them. So we give them something like chewing gum. They chew something that gets the gut going. We don't fill it up. There's less vomiting. This is really small because there are a whole lot of studies that have now been published. It's also really small because most of them are really small studies. One of the largest is actually an RCT we did in Bristol, based within the BRU but funded through an RFPB. Overall, there's a suggestion of reduced length of stay, but the studies are so small and the quality's poor and there's heterogeneity. I'm not convinced that chewing gum makes a difference. It's a lovely idea, but I think it's just judicious early feeding probably is the best data we've got. Another thing we've done, and this, I was trying to work out how to bring this in. We've used qualitative studies in lots of our preliminary work, drawing out people's narratives. And each one, when I present it, it feels somehow, it's small because that's the nature of qualitative work, but it was really interesting what people were telling us. So this is some work that a PhD student did around perioperative nutrition. So we got people who were having colorectal surgery, we went and interviewed them. One of the things was about information provision, that not only was it poor, it was often conflicting. One person said it was lucky I parked my car so near to the clinic or I wouldn't have been able to get all the information they gave me home. Which sort of sums up where we are. I was surprised that most people didn't report a problem of getting access to food on the wards. I didn't think that would be the case. Another thing I haven't highlighted here, which is one of the PhD students going forward, is both in PPI work and in qualitative work, people said, nobody knows what to tell me to do when they give me a colostomy or an ileostomy. I just stare at this bag and I'm saying, what should I eat? What should I do? And they say, oh, try this. And then the next person comes and says, try that. And they're completely different recommendations. And it really bothered people. So actually picking up issues that are important to people and trying to find out if there's an evidence base to inform it, or if it is just trial and error, is important. Epidemiology and health services research. So again, how can I do justice to so much in so little time? Say a little bit about life course, something about clinical trials, a bit about examples of causal inference, and then the development of cancer research here. So clearly ALSPAC figures and is now the flagship study for a lot of the life course work. It's not the only study. There are lots of others. Other historical and prospective cohort studies, also historical trials. So we did some work in the BRU doing some feasibility on a folate trial done. It was Marion Hall's PhD in MD in 1966. She gave pregnant women folate tablets at booking. We wanted to see if it left an epigenetic mark, because if it did, we thought, well, if that's there, then there's a justification for going back. And can we actually find these people? And yes, we found them, not as many as we'd hoped, and there is a suggestion of an epigenetic mark. So it's us using the feasibility capacity of the BRU to do the work that allows us to then apply for large grants to do proper follow-ups. And also large prospective studies that this was set up by Mike Kramer, but Richard Martin here has done a huge amount following up. This is a breastfeeding study in Belarus, and I'll refer to it in one of my examples. Clinical trials was a big part of what Bristol has done. I'll refer to one that as a favourite of mine, diet and reinfarction trial, just as an example. But actually the development of large trials such as the PROTECT study, which has been asking a question about how we treat people with prostate cancer, but on the back of that building whole research programs, bolting in some nutritional data, doing small randomized studies of diet and so on into that has been really powerful. And also we've done other smaller clinical trials, and one that we worked on, Fish and Mood, I'll use as an example. So this is a bit of history. This is diet and reinfarction trial. It was a trial run between 83 and 87, around 2,000 men across the southwest South Wales. This is run by, PI was Mike Burr, who was at the MRC epidemiology unit. And it was really striking. At 2 years, there was a 30% reduction in all-cause mortality for those that were given fatty fish advice. And they had a little fatty fish cookbook to help them do the job. And they'd made a decent difference in terms of changing fish intake. And the great thing about fish as an intervention for advice is it tends to be a fish-meat substitution. When you tell someone to eat low fat, you actually increase their fruit intake, you make a whole series of other changes. So it's quite a discrete change. The disappointing thing was when we did the follow-up, by which time around 1,000 people had died, so the longer-term follow-up, dietary compliance had slipped away, but the benefit had gone too. And actually now it's very unclear with the fish and heart disease story. A subsequent trial attempting to replicate wasn't convincing. In fact, if anything, went the other way. And other trials of fish oils as a supplement don't seem to show things. I think the other thing is that back then you could expect to do a trial in 2,000 people for 2 years and potentially get a result. You wouldn't get away with that now. Given that we have so many effective treatments, you'd be talking about tens of thousands potentially over 5 or 10 years. Enough about DART. It's just a... this is one of those slides about maybe epidemiologists getting smarter or not believing their own data. This is some early life data. This is a trial that was published in the BMJ. It was calcium intake in pregnancy and offspring blood pressure. And their main effect was null. But what they did see was this effect, a reduction in blood pressure in those that were heavier. And they made a big thing about that. We went back in ALSPAC data and tried to replicate it. It's not quite the same thing. It's not a trial. The dose isn't quite the same. It just isn't there. Epidemiologists, along with everybody else, we can be guilty of sometimes believing our own data. This is about being clever, and this is work that George and others led on. This is breastfeeding. So breastfeeding is something that is socially patterned. The women that breastfeed, certainly in the UK, will be different in lots of other ways. So we have a social gradient in the ALSPAC cohort. People who breastfeed have children with lower blood pressure, lower BMI, higher IQ. If you go to Brazil and plot us, you find there's no social gradient. You don't see any association with blood pressure and BMI, but it's still there for higher IQ. And then when you actually have the trial evidence, the trial I mentioned, the Probit trial, same thing, no effect for blood pressure, BMI, but it's there for IQ. So epidemiologists, I think, when they were setting up EPIC, they were right about heterogeneity, but what they should have been talking about was heterogeneity of confounding structure. And it's always surprised me about EPIC that they don't actually go and specifically look at that, to ask themselves, if I can see associations, let me see if there's a difference in the confounding structure, and is it there across those strata of confounding structure. So finally, in population science, cancer work. So I've mentioned PROTECT and the prostate cancer program, which, if I'm right, came out of, if my memory's right, came out of a series of commissioned regional reviews that then built Bristol's interest in prostate cancer. And we've also now got a CR UK program, which sits alongside George's MRC unit that Richard Martin leads, applying causal inference approaches. We've also started building clinical cohorts. So again, I'm shamelessly reporting the things I've done. This is a head and neck study we've built. But head and neck 5,000, and yeah, one of the themes here is about naming of studies. Don't put a number in your study, because there was a thing when we were having our second or third recruitment crisis of whether we're going to have to rename ourselves Head and Neck 500. We did actually get to 5,500 people, but it was a struggle. So yeah, don't put a number or a date in the title of a study. But this is now a resource for actually looking at people with disease observationally and looking forward. And this is something we've forgotten to do. We have cohorts in healthy populations, but actually looking at the groups with disease, describing them carefully and following them through. It got discredited because someone would, with my sort of level of OCD, would have written down everything they'd done for 30 years, and they'd write, this is my experience of this. And most of it was archaic. It was what someone did, that they were treated in a different way. But actually knowing what happens, if someone comes to you, what are my chances of this, is really important. So I think one of the things for the future is to actually, we have a strong track record of running cohorts, but thinking where we can establish clinical cohorts that sit alongside the general population cohorts. Also, this has contributed to some genetic work. We're part of an IOC-led consortium, interesting finding around SNP on chromosome 6, potentially related to oropharyngeal tumours, maybe related to HPV infection. And then, last but not least, is the nutrition behaviour unit. So, some pictures I lifted off the web. This is Peter Rogers and Jeff Brunstrom. This is, I think, reasonably up-to-date summary of their expertise, research interests, human appetite and weight control, food choice, dietary effects on mood and mental performance, psychopharmacology of caffeine. The way I characterise what they do, which I hope isn't going to upset them, is they do experiments on psychology students trying to learn about the way people eat. So they torture them with bowls of soup that never empty to see what affects appetite. What's fascinating is if we can harness those ideas and bring them into clinical practice, is the way that diet and appetite are shaped in a diabetic the same as it is in a psychology student? I'm not sure if it is qualitatively and certainly not quantitatively. And can we use this understanding of how people eat to develop interventions which are more effective? We have worked together over the time. This is a trial that, this blurred image is actually, that's me, this is Peter, this is David Goodall. This is a fish oil trial that we did on around 200 people. We got people with mild depression, tried them with this mixture of fish oils for 12 weeks. And actually, that was our trial. That was another one. That was actually the other DART trial that had a sub-arm around measuring and changing stress. And so they'd measured stress in relation to it as an outcome, and they'd had a fish oil intervention. But there wasn't much going on. So we've been quite good at showing things don't work. More recently, we've been able to apply the capacity we've had in the BRU to do policy relevant reviews. This is one that we did with, again with Peter, looking at the role of low energy sweeteners and actually pulling together, which for me was a revelation, trying to pull together animal data, short-term feeding experiments, long-term observational data, and clinical trials. And the overall suggestion that we came, and I'm reasonably confident about this, is that low energy sweeteners, they're not going to make you suddenly thin, but on balance the evidence suggests that they will reduce your weight rather than the other way around. It's just that if you're heavy, you'll tend to drink the diet whatever drink. And this is a rough idea of the sorts of things that we're bringing in. So maybe if we can compensate for reduced portion size. So I've mentioned about the sweeteners. Can we improve weight management using the models that are developed in experimental psychology? Is there carbohydrate-specific hunger? So it's harnessing our pipeline of ideas and bringing them into doing clinical studies, which has been what we've achieved over the time with the BRU, but what we're hoping to do with the BRC. So my past, present, and future. So my summary of the past. We had various groups doing work on nutrition, and some of them did clinical nutrition studies. And these were non-trivial. The studies I've reported were large, important trials. They tended to be specific projects or add-ons to larger projects. And there was collaboration on these projects, but it wasn't something that was ongoing and bigger than that. I think what the BRU brought us was coordinated support, dedicated methodologists lined up to do this. It brought clinicians alongside these methodologists. We, for the first time, had something that was visible for clinical nutrition. Instead of collaborative projects, we had a collaborative program. And we got to the point in many areas now where instead of someone having an occasional clinical thread or something they'd done for a while, most people have a clinical nutrition thread to their research portfolio which is ongoing. And we've begun to develop a training program. So what do I think the future might... so this is just a schema of our pipeline. So population science is an important part of what feeds into what we do. But so are the clinicians, the ideas, the questions they ask of us. The work done by physical activity in diet, experimental psychology, and others I haven't had much chance to mention. Working with Jeff Holly's group, they've made contributions in terms of not just analysing our samples, but suggesting ideas. So some of the work that we're doing now on whether we fast people when they have their chemotherapy was triggered by discussions with Jeff. So it's more than just this. It's trying to bring in a whole series of things and then feed it through. So we're supposed to sit at this translational gap. We're supposed to do the exciting studies, take it from the bench to the point where we can hand it to the trials unit to do the definitive trial. What would I like the future to be, or what do I imagine it might be? I think we need something that says we're a Bristol Clinical Nutrition Centre. But I would say that, of course, but I think something that is more visible, that says, when you go on a website, you say Bristol Clinical Nutrition, this is what it is and what it feeds into. It's strange. We started from large trials to go to now doing smaller scale studies. And it will come back to doing the large trials, but over the next 5 to 10 years, we need to be doing large trials, answering important questions. And I've argued that we should be thinking about important clinical cohorts. We've set one up in paediatrics around children with Down syndrome, where we're following them up from birth, trying to get samples, looking at how feeding changes, various measures. But I think it's a real challenge to think where we could set up cohorts. It would be great to have a trials unit here with a nutrition special interest, which was a go-to, so we strengthened our pipeline and strengthened our training. Dietitians are an untapped resource. There are not many of them that are doing clinical research. And providing them with the opportunities to learn about research, to engage with it, to make it fun, to make them convinced they can make a difference. If we could build a cadre of clinical academics who are dietitians, we could transform clinical nutrition in this country. So what would I love in 5 or 10 years to be looking at? Bristol, recognised leading international centre. I don't think people quite recognise how good Bristol is, and I think we can be so much better if we do the right things over the next few years. It's trying to thank everybody. I mean, this is 20 years of things that I've been involved in, the last 5 years with the BRU, which I think Julian and I both said has been the most rewarding period of our research lives. It's just been fun. And so many people, difficult to name everybody. I feel like someone who was on like Radio 1 who has this list they have to run through. So if I've not mentioned people as I've gone along, it's been done in partnership and a pleasure to do. And these are 2 bridges. This is actually Clifton Suspension Bridge, and this is the Alta Brücke in Heidelberg. Thank you.